we talked about mutations, right? And we mentioned briefly about the mechanism for repair. Okay. Now, how the mutations get repaired? Well, pretty much they being recognized. Okay. Here by the disturbance in force. Oh, sorry, the double strand. So DNA, when DNA has mutations, there is no proper pairing, no hydrogen bonds are formed. So you see something like this, a little bubble. There are no bonds because pairing is improper. Say you have G here and G here. Right, so basically, depending on, it doesn't really matter what caused that lack of proper pairing, lack of hydrogen bonds. One of the very common mechanisms is the nucleotide excision repair. Okay, so what happens is the enzyme called nuclease makes cuts on the sides of the mutation. Okay, like here. Then the enzyme that you've already encountered, called helicase, removes the fragment, physically holds it away. So now, instead of the mutation, you have a glaring gap. Next, DNA polymerase 1 fills the gap, and DNA ligase stitches the ends. Does that make sense? Something like, you know, when you fix something and you realize that, like, I don't know, you have furniture at home and you have a piece of furniture that's broken. Instead of trying to repair it, you throw the entire thing away and buy the new one. Okay, this is called the excision repair. And for the mutation that we've discussed last time, the thymine dimers, this repair happens in the dark. Well, this system works, it doesn't, let me put it this way, this system doesn't require light. It works in the absence of light. However, the major cause of thymine dimers is the UV light. Yes? New but matching. Does that make sense? Now, when we are exposed to UV light, we very frequently are outside and there's plenty of regular light. Regular visible light activates the enzyme that is called photolyase. Photolyase binds to the mutated fragment of the DNA and breaks down the covalent bond between two thymine dimers. So if we want to compare to that furniture piece, you have a broken furniture piece and you fix it, instead of throwing it away and buying a new one. Does that make sense? Now this is called photoreactivation, and this one requires the exposure to the visible light. So you see, one will work without light in the dark, another one would require the light exposure. Does that make sense? Now, how do we recognize that certain chemical is a mutagen? There are two ways. One, we can apply this chemical to the wild type microbes and then search for the mutated phenotype. Does that make sense? However, you gotta appreciate that, for instance, those plates that you have, any of those, among those colonies, you have a huge number of mutants. You just don't know what phenotypical changes are there. Maybe some biochemical alterations or something like that. But your populations, the total populations, are very heterogeneous. You understand the word heterogeneous? Does that make sense? Although we are all people, we are very different. Although they are all microbes of the same species, they are very different. So what we need to do, we need to select for the population that is homogeneous in one trait. Comparing to people, we would select for everybody with blue eyes or everybody who is blonde. Does that make sense? Just one trait. Got it? Now with bacteria it's really hard, as you can imagine. But there is a way. You can select for so-called auxotrophic mutant. What is auxotrophy? What is auxotrophic? Auxotrophs need a very special chemical to be present in the medium. Does it make sense? Say one more time. Say again. Specific nutrient. Yes. Yes. So, for example, in this example, I'm going to talk about auxotrophic mutant that requires the amino acid histidine. Okay, and we're going to call this Salmonella. So the microbe is going to be Salmonella typhimurium. Okay, so here's how we identify it. We have a complex medium, okay, that supports the growth of all different types of microbes. And obviously complex medium contains histidine. So we take a little velvet pad, press it against the colonies on the histidine containing medium, and then press this pad on two different plates. This plate carries the medium that has histidine. This plate carries the medium that does not have histidine. Does it make sense? All different kinds of Salmonella are going to grow on the histidine-rich medium. But medium that lacks histidine will prevent auxotrophic mutants from growing. Does it make sense? We can now compare the colonies on the histidine-rich and no histidine medium and identify the one that can grow with histidine but cannot without it. Does it make sense? We pretty much exclude histidine negative mutants and then can identify it. That makes sense how we identify it? So we finally have the strain of Salmonella that is histidine negative, does not produce histidine. It's mutant. Does it make sense? Now, our presumption is if the chemical that we test has mutagenic properties, one of its effects will be that this microbe will revert to the wild type phenotype. The mutation will be reverted. Does that make sense? Does it? That's okay. So mutagen will cause the reversion. Because it's also a mutation. For this guy, being histidine negative is new normal. Does that make sense? Can this reversion happen spontaneously? Yes. there are always mutations, and there can be 2, 3, 5, I don't know, 10 bacteria that actually came back to their original phenotype. So here is how the experiment is set up. The Salmonella typhimurium strain that is histidine negative is exposed to the mutagen or not. This is control. So in the control, you're going to see very few natural revertants. If this chemical, this mutagen, actually causes mutations, increases chances of mutations, has mutagenic potential, the number of revertants in the experimental plate will be higher than in the control plate. Does that make sense? Does it? Okay, good. Because that's the main important point here. Okay? So this is how you test for the ability of certain chemical. Now, what if the number of colonies in the control plate and experimental plate, on average, of course, because we're going to have a lot of plates, on average, the numbers are the same. What can you say about the chemical? Is it mutagenic or not? If the number of colonies, no, right? Because its mutagenic effects are no different from natural mutagenesis. Now, if you have two chemicals, and one increases the number of revertants by 10-fold, and another by 100-fold, which one is more stronger mutagen? 100-fold, right? The one that increases number of revertants by 100-fold. There's something that some compound sorry, something there that I never talked about. Look at the experiment, some experimental treatment that I avoided to talk about. Yeah, why? Why rat liver extract? What is the liver function? Among many others. Huh? Yes, but it's not relevant for this one. What does it do to various chemicals? Huh? Filters and converts. Okay? It chemically modifies things like toxins, okay, or drugs. So red liver extract is added there to model the potential effects of liver on the metabolism of the mutagen. Does it make sense? Because chemical by itself may not be extremely mutagenic, but when it is processed by a liver, it can become so, and vice versa. Does that make sense? Now, if we have positive results for mutagenesis, does that mean that we immediately jump to the conclusion and say we have to ban this chemical from everywhere? No, but now we have grounds to proceed and test it on mice, dogs, knowing what we look for. Okay? Which chemicals are potentially mutagenic? So like you test the chemical and it like causes, I don't know, 1,000-fold increase in mutagenicity. You probably don't want to put it in a hand grain. Okay? Does that make sense? But I can tell you can isolate a ton of chemicals from gas that are potential mutagens. But you're not going to ban gas in the country. What you're going to do, you're going to say inhalation or, consumption may lead to blah, blah, blah. And as they put, according to California state law, some of the chemicals are potential carcinogens. So you warned, now it's your decision whether you're going to inhale the gas in vast amounts or not. Does that make sense? Mutations are the great way to gain genetic diversity. The only problem is that they happen infrequently. So on a bigger scale, when you have bacterial cell dividing, daughter cells are going to have pretty much the same DNA. Right? So there's a huge chance daughter cells are going to be identical to the parental cells. Make sense? But if you think about eukaryotic organisms, especially the ones that employ sophisticated sexual reproduction, like mammals, fish, amphibian, whatever, okay, where you have male and female gametes, you have a good deal of genetic diversity just because of the genetic exchange, mixing the genetic information of both parents, right? Bacteria do not have sexual reproduction whatsoever. So they have evolved, the result of evolution in them are three major mechanisms of so-called horizontal gene transfer. Which means these mechanisms convey genetic information and exchange genetic information between cells that are not related to each other as a parent and a child. Although, however, you have to appreciate that in a bacterial population which divides like every 30 minutes, it's like rabbits. Very soon you don't know who's father, who's child. If you ever saw a good rabbit herd, you know that incest is a norm there. Now, these three mechanisms are transformation, when naked DNA is taken into the bacterial cell and either incorporated as a part of the bacterial chromosome or is maintained in a form of extrachromosomal plasmid. Transduction, which employs bacteriophages, the type of microbes that we didn't talk about, but we will have a chance to talk about them today. Also leading to the incorporation of the DNA into the chromosome. And finally, the bacterial sex conjugation, direct exchange of the DNA between two live bacterial cells. Does it make sense so far? Let's talk about transformation first. Transformation was discovered in the experiments of Frederick Griffith. As usually, I don't remember the year, 20th century, first half. Griffith was working with the gram-positive microbe called Streptococcus pneumoniae. You know it from pneumococcal vaccine. Okay, so he worked with two strains. The non-pathogenic rough strain did not have a capsule as a virulence factor, and therefore did not kill mice after inoculation. Mice survived the inoculation with non-pathogenic strain. Pathogenic strain, the S smooth strain, had gene for the capsule, was encapsulated, that was the main virulence factor, and although it is not shown in the picture, pathogenic strain successfully killed mice. So that's our initial data, right? So first question that Griffith asked, if we inactivate, if we kill the pathogenic strain by boiling or irradiation, in this case it was heat, boiling, if we kill the pathogenic strain and inoculate dead microbes into the mice, what's going to happen to the mice? It turned out that heat killed pathogenic strain did not kill animals. Are you keeping up? So far what we have, live non-pathogenic doesn't kill. Inactivated pathogenic doesn't kill. And then Griffith, in the attempt to identify how pathogenicity can be maintained, transferred, and so on, he mixed the live non-pathogenic and the dead pathogenic strains. Okay? And inoculated that mixture into the mouse. To his surprise, mice died. Some kind of a factor that was still present in heat inactivated pathogenic strain was able to confer pathogenicity to the non-pathogenic one. Moreover, Griffith managed to isolate the pathogenic strain from these mice. Does that make sense? So you can show it this way. If you have non-pathogenic rough strain, okay, and you have heat inactivated smooth strain. I'm going to put the circle around it so it says heat inactivated. Okay, dead. Does that make sense? And you mix them. And what you get is the result is absolutely perfectly live pathogenic S strain of the strep. Initially, Griffith thought that the pathogenicity was transferred by proteins. But eventually it was demonstrated that it was a DNA that conferred the genes of pathogenicity to the non-pathogenic strain. Essentially, what happened? The cells, I'm gonna picture non-pathogenic strain as a blue cells, okay? So blue cells of the avirulent non-pathogenic strain, and these are virulent pathogenic. Are you keeping up? Okay. Now, pathogenic strand gets inactivated. It's dead. When it gets inactivated, cells disassemble, disrupt, okay, and release DNA. Non-pathogenic strand takes this DNA in, and when genes in that DNA get expressed, especially genes that control the capsule, it becomes now pathogenic. Does that make sense? Now, this ability of a microbe to take in the naked DNA, most likely from the donor cell that died some time ago, right? Because where else can you get bacterial DNA? When bacterial cell explodes, it releases the DNA, so donor is dead. Recipient is very much alive. That ability to take in the DNA is widely used in biotechnology. Because when we refer to this, you will notice that, question? That, of course, that DNA that's taken in can be incorporated in the chromosome. But very frequently, that DNA remains in the form of a plasmid in the cytoplasm. Let's say you want to make a protein, an insulin, okay? You want to make insulin because you want to treat people with diabetes. synthesizing insulin using conventional classical organic chemistry will make it probably the most expensive drug on earth. Instead, what you can do, you can take the gene for the insulin and stick that gene into the plasmid. Then you can mix the plasmid, the DNA structure, with some bacterial cells, like E. coli, that grow readily. Then you apply certain type of a shock. It's pretty much a thermal shock to those cells. So cells take up the plasmid. Does that make sense? Then, when you will grow the cells, gene for the insulin on that plasmid will be expressed, and the insulin protein will be produced. Does it make sense? Now, you have millions and millions of bacterial cells before the transformation. Will every one of them take the plasmid in? No. How can you tell apart the ones that have plasmid and the ones that do not? You add some selective trait to the plasmid, and usually it's antibiotic resistance. Only cells that have plasmid in them will be able to survive on the medium that contains antibiotic. Does that make sense? That's a brief overview of the actual practice that you can do in the lab, or that is done in the biotechnology plants. And a lot of proteins, a lot of drugs, as far as I know, human insulin is produced exactly this way. They have gigantic fermenters in which they grow, I don't know about tons, but hundreds of liters of bacterial culture. And then they collect this bacterial culture, process it in a certain way, culture that those cells constantly churn out insulin, okay? So you have a ton of culture with insulin in it. All you need to do is to purify it, and it's a pretty straightforward process. Does that make sense? That's how it works. That's transformation. Yes.
So what you're saying is basically that bacteria will use dead bacteria in order to evolve?
It will not use the dead, but it will use genetic material from the dead cells.
So it doesn't have to be the same bacteria?
Great question. No, it doesn't. And that's what is scary. Because if you have a plasmid that confers antibiotic resistance, and this plasmid is somewhere in the environment, okay, this plasmid can be taken up by cells of other species. If you initially started with the vancomycin resistant gene in, I don't know, Enterococcus, what you can end up with is your entire hospital bacteria will be resistant to vancomycin. Does that make sense? Does that make sense? Plasmids are probably the most notorious way of spreading the antibiotic resistance through the population. Okay? That is really scary. Thank you for the great question. A what?
It's like a superpower taking like dead bacteria.
Well, if you want to,
Getting stronger.
if you want to talk in these terms, imagine that Superman kills Hulk. I don't know, eats Hulk, or eats its liver or something, okay? And now, in addition to Superman capabilities, he also has Hulk capabilities, okay? Transduction. For some reason, I removed those models over there. If you could help me, there are models behind you. I can unchain myself. If you could just give them to me, I want to show you something here, okay? Thank you. Just in any order, okay? So transduction, thanks, Brooke. Transduction is mediated by so-called bacteriophages, the evil-looking alien spaceships that are viruses of bacteria. So those are viruses, acellular not even organisms, acellular structures consisting of proteins and nucleic acids that can infect bacteria and replicate in them. Okay? The very brief summary of what transduction is when bacteriophage carries fragments of bacterial DNA between bacterial cells. Bacteriophage carries the fragments of bacterial DNA between bacterial cells. Now we're going to talk how they do it. For this, I'm going to use the model and I'm going to show it to you on the camera. Okay, so this is bacterial cell. Okay, follow? Bacterial cell. Unfortunately, this is not recorded. So, listen, the sound is recorded, but this is not. Bacterial cell, the phage attaches to it. You see that little dude up here? Okay? That's the phage that attaches to it. Now, when phage attaches, it doesn't get all the way in. Instead, it inserts its DNA. That little brown stick that you can see in the cell is the phage DNA. It injects it like through the needle. Kinda, sorta, yeah. Mosquito doesn't bury itself under your skin, it just penetrates with the proboscis. Okay? Same here. It injects only DNA. So here you can see the DNA is all the way down, right? That little brown stuff. And then, now you may understand why I love viruses so much. It hijacks the cell, which means DNA starts to use bacterial cell machinery to replicate itself. It's actually a hijack. Okay. So many copies of DNA are produced. Remember, DNA consists of genes, right? And those genes, phage genes, start to get expressed. And when they get expressed, they produce proteins, phage proteins. And those phage proteins have an amazing capability. They can self-assemble into the phage particles, like self-assembling Lego. Okay? So eventually, your bacterial cell becomes full of phage particles. Exactly, and then the cell goes boom. When those phages leave the cell explodes. Cell lysis. This is called, let me switch back, lytic phage. Okay? It lyses bacterial cell after its replication. Does that make sense? Now, when bacterial cell is lysed, what conceivably would happen to its DNA? Well, that can be an outcome, absolutely. I mean, the integrity of the DNA, what's going to happen? Most likely, it's going to get all chopped in pieces, right? So now you have hundreds of phage fragments, phage particles, phage units, hundreds of phages in the very closed quarters, packing themselves, incorporating DNA. By accident, one or two phages can pick up bacterial DNA instead of theirs. Does that make sense? And they will leave the cell, but they carry not their DNA, but bacterial DNA. Does that make sense? If they carry bacterial DNA, are they infectious? No, because they're not normal anymore. Does that make sense? They still can attach to a different cell and incorporate the DNA into the different cell. But they will incorporate not the phage DNA, but DNA from that dead bacteria. Does that make sense? So they will transfer fragments of DNA from one cell to another after the lysis. Does that make sense? So the process that we just learned is called generalized transduction. When phages destroy the cell, accidentally pick up the fragments of bacterial DNA and move it to another bacterial cell. Okay? Some phages have an interesting capacity. They don't kill the cell. Instead, their DNA gets incorporated into the bacterial chromosome. Such phages are called lysogenic. That's the word, lysogenic. So you can imagine, lysogenic means that they can produce lysis, but not right away. This phage fragment, which is sometimes called a prophage, is incorporated in the genome and sits there for a while, similar to some human infections. Can you think of any? So when this prophage sits in the genome and sits there for a while and nothing happens, yeah, like dormant viruses, okay? Mechanistically, you know, like herpes, right? Well, herpes has different mechanism, okay? But HIV incorporates its DNA into the human genome. So HIV is like human phage. It's all herpes. Shingles is a type of a herpes virus, but they don't incorporate the genome. They just go absolutely sleeping mode. Okay, but it's kind of it's sort of a latency. Does that make sense so far? At some point, the environmental triggers will signal the prophage to cut itself out, cut itself from the bacterial chromosome. The cutting, the excision, is always imperfect. Any idea what it means? I mean, what it means, the excision is imperfect. Yep, cuts out some of the bacterial DNA. Like, doctor, instead of amputating the finger, amputates half of the hand. Just by accident. It was imperfect. Okay?
Pretty big accident.
I know. Hmm?
Pretty big loss.
Yeah. So, well, they can do that. I mean, lawsuits. So, when they cut out some part of bacterial DNA, can be excised as well. Does that make sense? Then the normal phage synthesis and assembly continues, okay? This time, phage packages not just phage DNA, but also a fragment of a bacterial one. Does that make sense? Yes. Oh no, it excises itself. It has pretty much a chemical machinery. It folds in a certain way. There are some organic reactions happening. It comes out of the chromosome. Does that make sense? At which step? Give me number of step. Number 2? Yes. Bacteriophage? Absolutely. It just sits in a chromosome, not doing squat. Okay? So far makes sense? So this generation of phages will have both phage DNA and the fragment of bacterial chromosome. And when it performs second round of infection, it will incorporate phage and bacterial chromosome and the fragment of bacterial chromosome now. That makes sense? This is called a specialized transduction. I can tell you more. Sometimes simple incorporation of the phage DNA like this can cause changes in a bacterial phenotype. If I remember correctly, Corynebacterium diphtheria, you know the disease diphtheria, right? The diphtheria toxin is encoded by the phage, which incorporated its genome in the bacterial chromosome I don't know how many 100,000 years ago, and since then the microbe is pathogenic. If you artificially cut this part of the genome out and stitch the microbial DNA back together, Corynebacterium loses its infectivity. It's not pathogenic anymore, but it can replicate perfectly fine. Does that make sense? Yes. Yes. We know the genomes of huge numbers of bacteria, honestly. I think that now we know the genomes of virtually every important human pathogen there is. And with availability of fast sequencing, high throughput sequencing, a lot of scientists now don't even bother about some sophisticated approaches. They just brute force it. Oh, we've got a microbe. Oh, let's just sequence it and just see what we've got here. It's great. It really, if you can afford it, that's perfect. Does that make sense? The transduction part. Now you can imagine that phages can move it right and left. Now, the actual sex, it's not as exciting as a human sex, not as messy. The conjugation, I'm not kidding, in some papers, like in papers in microbiology, you sometimes can see the phrase conjugation or bacterial sex. It is considered to be as close to the sexual reproduction as possible. So in this case, you have a donor and the recipient cells. Okay? Donor cell, which is also called F positive. F stands for fertility. Donor cell possesses the fertility plasmid. This plasmid not only has the capacity to get transferred, it stimulates the formation of so-called fertility or F pilus. When this pilus is formed, these two cells can communicate. F plasmid gets copied. Copy of the plasmid is transferred to the recipient cell, and now you have two F positive cells. Does that make sense? Let me ask you this. If you have a population of bacteria in which there's hypothetically, one cell with a fertility plasmid, and you just let this population grow, what's going to happen in the end? They're all going to be fertile. They're all going to have fertility plasmid. Now, honestly, I couldn't care less about fertility of microbes, but the problem is that plasmid, that F plasmid, can carry other traits. And as humans, I wouldn't be even concerned about virulence factors, but I would be concerned about the antibiotic resistance. And ultimately, if you have antibiotic resistance gene on the F plasmid, it's going to spread through the population like forest fire. The entire population will eventually acquire antibiotic resistance. And that is why, for instance, if you have just plasmid, not fertility plasmid, that possesses the antibiotic resistance, then of course transformation may help communicating that plasmid between the cells. Does that make sense? But if it's F plasmid, then you're screwed. Make sense? On this particular part, any questions? Okay, that is one of the most important mechanisms for antibiotic resistance transfer in the populations. The scary part about it, as humans, we, most of the time, vast majority, mate with the same species. Some people don't, but that is considered to be a perversion. It depends on the state. In Louisiana, I think in Louisiana it's legal. Okay. We looked it up once when we were in Louisiana. Oral sex is illegal in Louisiana. Well, the closer species, if we talk about higher mammals, say we take the tiger and the lion, they will produce a progeny that will be infertile. Humans and a monkey would probably work. I don't know if it'll die. It may. I don't know. I don't really know if there are any recorded events like that. The what? No, I don't think so. I don't think so. Yeah. We had a joke, an old joke, nerdy joke about, scientists reporting the first attempt to produce a hybrid of the giraffe and an elephant on the scientific conference, and somebody from the audience says, "So, what's the result? How was it?" I say, "Well, I'm not sure about the result, but animals really enjoyed the process." Well, I mean, pick any two. Pick any two. Like, what's going to happen if you hybridize the snake and the hedgehog? 6 feet of a barbed wire. Now, other things that can happen to the F plasmid is that F plasmid can get incorporated into the chromosome. Okay? Now, if things can get incorporated, they can get excised as well. And what I told you about excision, it's imperfect. Okay? So when this plasmid gets excised back out of the chromosome, it can now contain a little fragment of the bacterial genome. Does that make sense? That makes sense. You put F plasmid into the chromosome, then it gets excised from the chromosome. But what comes out is slightly different from the original F plasmid. So it's now called an F prime. plasmid. Okay? So this process is called high frequency recombination, sorry. So when recombination, plasmid gets in, gets out, gets in, gets out, gets into the chromosome, gets out of the chromosome. Every time there is a recombination part, there can be a fragment of the chromosome that is taken out on the plasmid. When such plasmid is transferred to a new cell, well, the new cell is called F prime cell, which is a fertility chromosome plus some bells and whistles that it stole pretty much from the bacterial chromosome. So that also increases the genetic variability in a population. Okay? The craziest part is that sometimes the F plasmid that's integrated in the chromosome attempts, well, it's not like physical attempts, but it has genes that send such signal. It will try to transfer the entire genome of the donor cell into the recipient, okay, during conjugation. Now, conjugation doesn't last forever. Pilus isn't forever. Pilus stays between two cells for a certain period of time. And usually that period of time is not long enough for the entire chromosome to get transferred. So only part of it goes into the recipient cell. Does that make sense? In one case, this pilus can stay formed for 10 minutes. In another for 20, 30, 40, 50, different times. Does that make sense? As you can imagine, during different period of time, certain parts of the genome are going to get transferred. So and the origin of transfer, the part of the chromosome that gets transferred first is always the same. So you can map the position of the genes based on how many genes got transferred during a certain period of time. Does that make sense? Yes.
So if the whole thing doesn't get transferred over, what happens with the remaining part that's left in the old cell? The polymers come in and rebuild it?
That's a great question. I would say it's probably, on the other hand, it's probably going to get destroyed because it doesn't assume the proper circular shapes, and there are receptors that recognize it and probably destroy it. Okay? Because it's a copy. It's not like you take the entire chromosome away and now we have cell without it. It's a copy. But I understand the remainder of that copy, what's going to happen to it. I would bet on the destruction. Whenever they need, yes. Okay? Now, imagine that it was 10 minutes. So first 3 genes got transferred. Then in 20 minutes, first 3 and next 4 got transferred. By analyzing which genes got transferred into the recipient cell over a certain period of time, you can map the bacterial genome using minutes as the measurement unit. Okay? So, for instance, if this gene, PABB, is at 40 minutes, it means that for that particular genome, I think it's E. coli, okay? For that particular genome, it takes 40 minutes to transfer the part of the genome that includes the gene PABB, okay? If you would compare this gene and this one will be closer to the origin of the transfer than this one. So you can compare their position in the genome, where they are. Does that make sense? Because so then when you manipulate the genome, it's easier when you know where the genes are. That makes sense? We talked a lot about how genes are regulated, right? Expression, regulated, genes are expressed at the given time, produce certain phenotype. But only once we mentioned the actual mechanism of the regulation of expression. Anyone remember that? How can we control which genes on the DNA get expressed and which aren't getting expressed? Huh? Not random. Well, splicing partially controls at the DNA level. DNA in the nucleoid. Does it exist as only DNA, or it is in a complex with something else? It is folded around what? Histone-like proteins. In the bacteria, it's histone-like proteins. Remember we discussed that when DNA is wrapped around them, it's inaccessible for expression. Does that make sense? When it is unwrapped, it's accessible. So that's one level of regulation, one level of control. Now we're going to talk about two more. The concept of bacterial operons and riboswitches. So operon is kind of a team of genes that are linked together, that are related together, because they are necessary for a specific task. Imagine, like when you build a house, you will need guys that know how to put down the roof, like make a foundation, I don't know, finish the basement, millions of specialties, right? So what you do when you want to build a house, when you just have a land, you don't go here and hire one guy and there and hire another guy. You call, Yeah, you call a company and they send the entire team. And that team knows how to build a house. If you will ask them to build a boat, they probably won't be able to do that because it's a completely different story, right? You see what I'm saying? So operon is assembly of genes that allow cell to do something. Synthesize amino acid. Use sugar as the source of energy. Does that make sense? It takes more than one gene, for example, to use lactose as the nutritional source. Okay? So those genes that actually perform that function, those guys in the team, are called structural genes. Now, what is the first step in gene expression? DNA has to be transcribed. First, it has to be transcribed. What is the enzyme that transcribes? RNA polymerase. Okay? During transcription, RNA polymerase produces RNA. You keeping up with me? RNA polymerase. Which sequence does it bind? Which part of the DNA does it bind to? Before the genes. If I will give you a hint, you will tell me right away. I want to torture you a little. No, that's translation. RNA polymerase binds something that starts. No, what? No, it's translation. No, that's replication. Okay, it promotes the transcription. Promoter. It binds promoter. Remember? The short fragment that does not encode for anything. It serves entirely for RNA polymerase binding. Okay? Here's the promoter in the operon. You can see that between the promoter in the structural genes the sequence that is called operator. That operator sequence will, well, I will have to make it anthropomorphic, will decide whether transcription is a go or no go. When you run a distance, you start from this, you stay on the start, okay? And you know, you have to cover this 100 meters. But if somebody would put the wall in front of you, that you cannot jump over, that you cannot walk around, you won't be able to cover those 100 meters. Does that make sense? So the function of operator is to put that wall, or remove it, or put it back, or remove it. That's where the wall is getting installed. Does that make sense? So essentially, the operator sequence is what controls the expression of the structural genes. Keeping up? Sometimes regulatory genes are getting involved, but not going to discuss how exactly they can get involved, okay? When we will discuss the operons, we're going to focus on the function of the operator. Deal? Operons were known, I wanted to say for centuries, no, not for centuries, but they were probably one of the first elements of genomic structure in the DNA that were discovered. They were known for quite a long time, maybe 50s, 60s, something like that. Okay. Riboswitches were discovered much later, not that long ago actually. And for some reason, they scare students. They think it's such a complicated issue. But before we start talking about them, can you name a switch? What is, do you know any switches? Huh? Light switch, yeah, perfect, light switch, excellent, light switch. Now, this is a fancy light switch, right? Do you know the switches, that mechanical switches, right? At home. Did you ever disassemble one? Or peeked inside? Well, you have, I know. If you haven't, I'm going to tell you, I'm not an electrician at all, I have two left hands, but if you look inside, you know, kind of flick it up, and inside something changes, like a little lever goes down and makes a contact. You flick it down, the lever goes back. So every time you turn the light on, some structure inside the switch changes. You turn the light off, structure changes again. Does that make sense? Usually, the simplest switch, which positions does it have? On, off. Right? Two positions. Riboswitches switches are the same. They have on and off. And they turn on or turn off. They can turn on and off transcription, or they can turn on and off replication. So far, does that make sense? We good? So far, we good? I'm not gonna continue unless everybody understands the idea of on-off. Yes.
Operons and riboswitches, is there any specific situations they're using? This is just general, like there's another piece of transcription.
Those are two basic mechanisms of regulation. Does that make sense? Like, they, for a specific trait, they may or may not be present. A particular gene may or may not have riboswitches. What I'm saying is that they can be present and they are common. Does that make sense? So the purpose, huh?
Their overall purpose.
Yeah, so if you read something and you see the word riboswitch, you know what it is. If you read something and you see the word operon, you know what it is. So with operons, I'm going to use a couple of examples. With riboswitches, I'm going to just explain generically how they work. So ribo, in which molecule they will exist? If it's ribo, another RNA, yeah, ribonucleic acid. Okay, riboswitches are special structures in the RNA. You can see the riboswitch here. Okay, and adjacent to it, you have another structure, the stem loop structure, that is called anti-terminator. That makes sense? Now, RNA reads the DNA during transcription. I'm going to highlight the DNA here, okay? And produces newly synthesized RNA. Does that make sense so far? Now, anti-terminator loop means that that same mRNA that DNA churns out already has a special structure that tells mRNA, well, quote unquote tells mRNA, not mRNA, quote unquote tells the enzyme, keep going, keep going. Now, there is some small molecule. Usually it's organic molecule that binds to the riboswitch. It's not the covalent bond. It's non-chemic, physically binds. When it binds to the riboswitch, it changes the function of the riboswitch and of the adjacent fragment of the RNA, converting anti-terminator loop into the terminator loop. When it happens, terminator loop serves as a signal for RNA polymerase to stop transcription and fall off. Does that make sense? So this riboswitch works for transcription. This one works for translation. The basic underlying principle is the same. Small molecule binds to the riboswitch, changes its structure and the structure of adjacent ribosome binding site. When structure of the site where ribosome binds is changed, ribosome cannot bind anymore, and there is no translation. Yes?
Since bacteria do both at the same time, does it only take one switch to turn it all off, or just sections of it off? We need a bunch of those switches.
Great question. I can answer. So your question is, does the riboswitch switches the...
Can it turn off the transcription and translation off at once, or can it do, or does it need two different switches to do that, since they do it at the same time?
Okay, that's going to be two different switches. That's going to be two different switches, because if you shut down the transcription, you still have the transcript. It may be short, it may be incomplete, but it is there. So it's going to get translated. Does that make sense? For translation, you shut down translation, period. Now, what can be those little molecules? Remember we talked briefly about quorum sensing. When bacteria produce chemical mediators that regulate, for example, the population density, that regulate pigment production, serratia. When they grow in the dense populations, they produce white colonies. When populations become sparse, colonies become more and more pink red. Okay? That's an example of the quorum sensing. Does that make sense? So that small molecule right here can be a quorum sensing product. Something that other bacteria release. So when they release that molecule, it diffuses into bacterial cell and stops them from making this, or that. Got it? For God's sakes, it can be antibiotic. It can bind the riboswitch, shut down the translation or transcription, and kill the cell. Seriously. So it's, by the way, I don't know about any clinically approved antibiotics that work this way. But I think at some point it may be the avenue for farm research. Now the operons, there are two types. The inducible operons and repressible operons. And before we move on with discussion of those, that's the last thing that we're going to talk about today. Before we move on, I want to remind you the basic principle of enzymatic reaction. Every enzymatic reaction starts with what? Substrate, which is converted into product. And what converts it? Enzyme, yes. So keep it in mind. At the bottom, substrate is converted into product. An enzyme does it. All right? Now, first, inducible operons. The lactose operon. Lactose operon, its structural genes encode enzymes that are necessary for use of lactose as the nutritional source. Does that make sense? The structural genes are enzyme. They encode for enzymes that bacteria need to use lactose. That statement, is it clear? Structural genes in the lactose operon encode for enzymes that bacteria needs to use lactose. Okay? When lactose in the environment is absent, there's no lactose, do we need those enzymes? Why we don't need those enzymes? Why bacteria doesn't need them? There's no substrate, right? There's no substrate for the reaction when there's no lactose. Is that clear? Is that understood? There's no substance. Okay? So how can we prevent expression of those genes? We simply put repressor at the operator sequence. So RNA polymerase, although it can bind to the promoter, it cannot proceed with the transcription. Does that make sense? When lactose appears in the environment, lactose molecule binds to the repressor, and this complex dissociates from DNA. Does that make sense?
Yes, except for what is the repressor?
It's some protein. It's just a protein. Repressor protein. Okay? So when complex is dissociated, nothing prevents RNA polymerase from starting transcription and expressing the genes. Does that make sense? Now, when these genes are expressed, what is the result of gene expression? The ultimate result of gene expression. Which molecules? Polymers. Transcription, translation, proteins, yes, right? So those enzymes are proteins. What those enzymes here will do with the lactose? Huh? You'll break it down. And when you break down lactose, what happens to the levels of lactose? They go down, and as they go down, and say all lactose is used, what's going to happen to that complex? Yep, repressor will go back, and when lactose is all used up, the expression of the lactose metabolism genes will be shut down. So inducible, this is called an inducible operon. Inducible operons are regulated by the presence of a substrate. Does that make sense? Now, repressible operons, we're going to discuss them using tryptophan operon as an example. Tryptophan operon controls the genes that are necessary for the synthesis of amino acid, tryptophan. Normally, repressor is not bound to the operator, and RNA polymerase continues with the expression. Does that make sense? These genes are produced, and products of this gene expression, products of the proteins that are encoded by these genes, they make a metabolic pathway which ends up in synthesis of tryptophan. When tryptophan in the cell accumulates to certain levels, it binds to the repressor. The complex binds with an operator and transcription stops. When transcription of those genes is stopped, what happens to the levels of the enzymes that make tryptophan? They go down. What happens to the levels of the tryptophan? They go down. When levels of the tryptophan go down, what happens to that complex? It dissociates because there's no tryptophan. Repressor goes away from operator, and the entire system starts to work again. Yeah, bingo, that's negative feedback, okay? In both cases, it's negative feedback. So, repressible operons are regulated by the product of the enzymatic reaction. Does that make sense? Think about car making. Making of anything. Okay? Like when you make some kind of a equipment and sell it, if everybody bought it, you don't have a market anymore. So you're going to shut down your manufacturing. Does that make sense? That's repressible operon. I don't know, the mnemonic rule that you can use to kind of keep it as a second option. The word inducible doesn't have any P letters in it. The word repressible does. So repressible is regulated by P product. I use it often, that type of rules. Okay? Yes?
The operator act on its own without a repressor?
Oh, operator is just sequence of the DNA that repressor recognizes. So the absence or presence of the repressor, the binding of repressor, is what regulates the activity of the operon. Does that make sense? So basically, operons are structures above the level of a single gene that regulate gene expression. Riboswitches are structures not in the DNA, but in the mRNA that provide additional level of control. Does that make sense? Okay, good.